Welcome to our video, Protein Synthesis Made Simple. The story will start with DNA, which is a double helix or a long chain of nucleotides which contains genes. In this example, we chose this small section of the gene, and the DNA has two strands. In this case, we will have a template strand and what is known as a coding strand. And what we will generate by the process of transcription is a piece of mRNA, and that will be based off of the template strand. That mRNA will be processed, ultimately leaving the nucleus, heading out to the ribosomes in the cytoplasm, and their translation will take amino acids and begin building a polypeptide chain, which will ultimately fold and form a functioning protein. DNA, once again, is a macromolecule, and it is composed of nucleotides. And a DNA nucleotide is going to have a phosphate group. It is going to have a pentose sugar. And it is going to have a nitrogen-containing base. In DNA, those bases are A, T, G, or C. And then in RNA, we will sub out the T for a U. Here you can see that the nucleus is in the cell and it contains the chromosomes. And if you unwind or zoom into a chromosome there, you can see the gene. The gene will have the code for the protein. The gene is the code for the protein or the enzyme or the hormone. And you need to get the information from the DNA out. And you will do that by generating a molecule of mRNA. Once that mRNA is created once again, it will leave through nuclear pores and head out to the ribosome. These ribosomes can either be found attached to the endoplasmic reticulum, otherwise known as the rough endoplasmic reticulum, or free-floating in the cytoplasm. These will be your free ribosomes, and any protein made here will stay in the cell. And any protein made on these bound ribosomes will end up either in the cell membrane or exported from the cell in the case of a hormone. Transcription is the creation of the mRNA, and you will be using the template strand of the DNA and an enzyme called RNA polymerase II. And in this case, you can see that if the template strand has an A, the complementary base on the now newly formed mRNA will be a U. If there's a C, RNA polymerase II will bring over a G, and so on. Just like DNA, you can only build onto the new mRNA on the 3' end. Here you can see the bigger picture. And you will see the template strand, or the one strand of the DNA. The other strand is called the coding strand. And later on I'm going to show you that the coding strand is actually the gene, or is actually the instructions for the gene. Because you'll notice that the A here matches up with the A there. Now we know that RNA does not have any T's, so instead of a T you'll have a U. And if you follow along, G to G and A to A. And if this continues, you will see the pattern continues. And once again, what you just said, if you look at the coding strand, you will see that the bases there, basically mimic the mRNA, just instead of T's, there are U's. Here again is the example where the base of the coding strand, in this case the A, will match up with the A on the mRNA. T will be converted over to U, G to G, and so on. So the RNA polymerase II does need a place to start, and this area will be known as the promoter region. And in prokaryotes and eukaryotes, it's a slightly different story, but prokaryotes obviously are more simple, and eukaryotes will need a little more help. So in a prokaryote, the RNA polymerase II is going to bind to a promoter region and start transcription. It's pretty simple. You're going to find your promoter site and begin transcribing the mRNA. Prokaryotes, like bacteria, can start transcription and then begin translation basically at the same time. That is something that eukaryotes cannot do. We call this coupling. And in this case, the bacteria can generate proteins or enzymes very, very quickly. In this diagram here, you can see that the DNA is basically transcribing the mRNA and then immediately translating the protein. And here's your DNA strand right here. There's your mRNA heading directly out to the ribosome. So it's in essence one continuous story. In eukaryotes, however, the process is a little bit different and more complicated. You will still have a promoter region, but in this case, you're going to need some transcription factors. And you can see here these transcription factors are near the promoter region, and once there are enough transcription factors, the RNA polymerase II can begin to transcribe the mRNA strand. The mRNA contains all of the same non-coding regions that the DNA had, and it is unprotected at this time. So the first thing you have to do is put a 5' cap or a GTP cap, and that will be at one end, or the 5' end. And the other end will have what is known as a poly-A tail, and there will be many adenines making this poly-A tail, and that is on the 3' end. Once the mRNA is created and it is capped, it now needs to be processed. Now those sections of coding, or the coding regions, are known as exons, and the non-coding regions are known as introns. The introns need to be cut out, and they're going to be cut out using these SNRPs, and then the pieces will be spliced together using spliceosomes. So let's take a look at this diagram. Here you can see your pre-mRNA has a lot of extra bases, and in comes the SNRPs. They're going to begin to cut out these intron pieces. Then you need to take the exons, there's one and there's one, and you need to splice them together. So in this case now you can see that the mRNA is shorter and it does not contain any non-coding regions. Now that it's been processed and it's shortened, it's considered mature. This molecule can finally leave the nucleus through the nuclear pores and head out to the ribosomes. Once it gets out there, it's going to first bind to the small subunit, and once again, a ribosome contains a small subunit and a large subunit. And you're going to be looking for what is known as the start codon. And the start codon, it will be AUG. That's going to go on to code for the amino acid methionine. You can see the methionine, or met, is right there. You will see that there is an A site, a P site, and an E site in the ribosome. That's going to allow for molecules of tRNA to come in and eventually swap or add on the amino acids. The tRNA will bring amino acids to the mRNA on the ribosome. This will happen at the start codon, or AUG, and end once the ribosome reaches one of the 3 stop codons, and they are UAA, UAG, or UGA. Looking at the universal genetic code, the 3 stop codons are over here, and the 1 start codon is over here. This is the entire process all in one diagram. Go or start will happen down at one end. It will continue to translation, or adding amino acids to the growing polypeptide chain until a stop codon is reached, and once that is reached, everything falls apart. You will have your mRNA will go one way, the ribosomes will go another way, and the final produced polypeptide chain will also leave. Once it leaves, it will begin to fold, and when it folds, it will be ready to function. So once again, once the stop codon is reached, the ribosome, the polypeptide chain, and everything fall off, protein will begin to fold. This only happens if the conditions are within the protein or enzyme's normal or optimal range. If it is too hot or too cold, the folding process will be interrupted or may not happen at all, and the protein will be non-functional. So here's a cleaner picture here of the story. This would be or represent your stop codon. And once you reach that stop codon, everything falls off. Ribosome breaks up. Stop goes one way. mRNA goes one way. And the free polypeptide chain goes one way. And it's going to start unfolded. And it will begin to fold, assuming the temperature, pH, and other cellular conditions are in optimal range. So now, let's draw it all together as we did in class. If you look on the left, you will see your DNA, or the whole left side of the diagram is the nucleus. You have your template strand and your coding strand. Through the process of transcription using the enzyme RNA polymerase II, you will generate a piece of mRNA. After it's processed, meaning 5' cap, poly A tail, introns spliced out, snurped out, and exons spliced together, the now mature mRNA can leave and head out to the ribosome. Once it gets out and finds the ribosome, you will notice that the nucleotide bases are in packs of 3, and those 3 nucleotide sections are known as codons. Now, also out in the cytoplasm, you will find amino acids. They are represented by my square, my black square. And then you will find molecules of tRNA, which I have in green. The bottom portion there of the tRNA is known as an anticodon. That is going to allow for a temporary kind of junction when the tRNA joins up at the ribosome. So if you look, GAA, using your universal genetic code, is going to call for the amino acid glue, and that's going to be brought over. The anticodon and codon are going to form an association and hold it for just a little bit. Then the next amino acid is pro, and you can see that the ribosome moved down, went that way, and that tRNA brought the pro or proline to the amino acid and mRNA strand. The next thing that will happen, it's 2 things will happen actually. First, you are going to form a peptide bond that is going to allow your 2 amino acids to be held together. The tRNA that originally brought over the glue is going to now leave. It's going to return to the cytoplasm to eventually be called back again. This process will continue where the next amino acid lined up will be gly. That's going to begin to move its way to the ribosome. This tRNA molecule will eventually leave, and another peptide bond will be formed. There it comes. There it goes. That's on its way back. There's your new peptide bond. And this process will continue. It'll continue until you reach a stop codon, any one of these 3 stop codons. At that point, the whole story falls apart. So in this case, I use the stop codon of UAA. Once that is reached, ribosome goes one way, mRNA goes one way, the last tRNA goes one way, and then your polypeptide chain is free to begin its folding process. If the conditions are right, the polypeptide chain will fold and ultimately function. And here are some crazy examples of different kinds of proteins. You will never be asked to memorize or redraw any of these, but I put them there just to show how complicated the folding patterns can be. And hopefully you enjoyed our protein synthesis made simple.